welcome to video 12 of your IB biology course, looking at topic 2, molecular biology, and in this video we are looking at proteins, unit 2.4. Okay, so before we start, let's go back to that whole idea from 2.1 about monomers. And monomers being the single unit of our long chain molecules. Okay, so when we think of proteins, our monomer is our amino acids. Okay, so if we look here, we have the structure of one amino acid, the molecular structure, and here we can see our polypeptide chain. Now, our polypeptide is the peptide bond, so it means many peptide bonds. And we've got our individual amino acids, okay? Alrighty, so thinking about our amino acids. As I said, we call it a polypeptide chain because we have these peptide bonds that form between our amino acids. So here we've got two amino acids, we've got glycine and glycine that are joining together. Okay, you will see that is water required here or is water released? Okay, so you will see that water is actually released because the carbon and the nitrogen bond together. Water is released, which means it's one of our condensation reactions again. Okay, and a peptide bond is formed. Our polypeptide chain means many peptide bonds. Amino acids linked together by condensation reaction, as we just said. Two amino acids linked together form a dipeptide, two di. More than two amino acids linked together form what we call our polypeptide. This process of amino acids joining together, this condensation reaction, occurs on the ribosome in the cell. Okay, so one of your skills is drawing molecular diagrams to show the formation of a peptide bond. We will practice this in class, but it is one of your skills. Alrighty, so amino acids, all right, big group of diagrams here. Don't panic too much about them. Let's see what we're going to say about them. Now, an amino acid is a pretty small molecule, okay, with some characteristic groups of atoms that determine its chemical behavior. The R group is the only part that differs between every amino acid, okay? So the R group is here, highlighted in blue on these diagrams. If you look, the rest of the amino acid structure is the same in each of them. So these blue areas on these diagrams are the R groups. And you'll see all these amino acids have got different names underneath them. Okay, and that R group is the thing that makes them different, remember. Alrighty, an amino acid is a relatively small molecule, as we said. It's the R group that makes them different. So having a look at this here, showing us the structures. So we've got the amino group, we've got the acid. Now this is alanine, valine. You see how this R group is changing each time. Okay, so that is the R group that was changing. Alrighty, quick look back at condensation reactions. We've got our two amino acids joined together, condensation reaction, water is released. We go from two single amino acids to a polypeptide chain. Okay, so condensation reactions occur in the formation of polypeptides from single amino acids. Yep, two amino acids can link together to form a dipeptide and water. Many amino acids link together to form a polypeptide. We've already covered this. The bonds formed between two amino acids is called the peptide bond. If we repeat it, it just becomes clearer. Polypeptides, some great diagrams here. Just take a minute to have a look at them. So you've got these long chains of different amino acids. Each of those colours is a different amino acid, okay? And here we've got amino acids, and these here, okay? These are our R groups. So we've got 4 different amino acids here with the different R groups. And then bonding them together, so we're going the lilac colour to the pink colour, we've got our peptide bond, peptide bond, peptide bond, okay? And then that 3-dimensional shape, all of these things are all aligned. Remember, those proteins are quite kinked and scruffy kind of chains, okay? Not neatly laid out or anything. Okay, polypeptides. So these long chains of amino acids differ in many different ways, in length, in the order of the amino acid, and in the type of amino acids in the chain. So there's lots of differences. That's what makes a polypeptide protein, same thing. That's what makes all our vast range of different proteins, is the order of the amino acid, the length of the chain, and things like that. Alrighty, so polypeptides are created via these processes of transcription and translation. We will learn these in a later video, okay? Just trust me on this one at the moment. DNA has the code for which a polypeptide chain will be produced, okay? And this comes from reading genes. Again, we will go into this later. The polypeptide chain will then be folded into a specific shape. So basically, we get the chain, which is made from reading the DNA. Then bonds start to occur between atoms. So, for example, the hydrogen bonds here, that pulls it into what we call the secondary shape. And then the third level here is further bonds causing further kinks. So we have what we call our primary level, secondary level, tertiary level. We will look at this further. The shape determines the ultimate function of that protein. So think about it. We've got these different amino acids in the chain. Now, depending on the placement of those amino acids, that causes different shapes to occur. So you're looking at that oxygen and hydrogen forming the hydrogen bond. Now, it depends the placements of those in all the amino acids, which kind of determines different shapes, okay? And again, when we get to that tertiary level, the same thing. So the order of the amino acid determines the shape of the acid, and the shape of the acid, the shape of the polypeptide then determines its ultimate function. It's fascinating. Proteins are fascinating. Okay, a polypeptide is not a protein until it is folded into its correct shape to carry out its function. So that long chain of amino acids, all right, that is our polypeptide. It is not actually a protein until it's folded into shape, ready to perform that function. There are 4 levels of protein structure: the primary structure, secondary structure, tertiary structure, and then it's called ternary structure. So that previous one didn't quite touch on that previous slide. Alrighty, the primary structure of proteins. This is the linear sequence of the amino acids, okay? So that polypeptide chain. It's unique for each protein. The order of those amino acids will be unique for each protein, and that's what makes each protein each protein. Determines the function of the protein because it's the structure that then determines the shape and all those folds. All right, secondary structure. It can be an alpha helix or a contorted bed. The primary structure that have coiled, pleated, or folded is all due to these hydrogen bonds. Get an alpha helix coiled allows the protein to stretch and recoil, a bit like in keratin, a protein. Beta pleated, the protein is stretched and more rigid. Sheets make up fibrin. Okay, it's a bit like silk. All right, moving on to, we will look at each of these a bit further in class, so don't panic about this. Tertiary structure of proteins. All right, so we've got the secondary structure, which folds into a after the secondary structure, we now fold into what we call this globular shape. That line doesn't make much sense. Parts of the secondary structure are attracted then to other areas. So those weak hydrogen bonds have made that secondary structure. Now what's happening is other oxygens and hydrogens are being attracted to then bend it into like this globular structure. We'll do this with some hands-on material in the classroom, okay? But it makes what we call this globular shape. So you can kind of see the alpha helix happening here, but then it's been bent further. Typically, when proteins become functional, but this isn't always, okay? So usually at this stage, they start to become functional. Not always. Okay, the quaternary structure of proteins, so this is the fourth one. This is formed by two or more polypeptide chains. And it's the assembly of a cluster of protein chains as such, joined together to form what we call a functional protein. Now, as I said, we will go back over these in class, so don't panic. This is your first introduction to this. All right, so levels of this protein structure, the summary. Primary, that is that initial stage of our polypeptide chain forming, okay? Secondary, those hydrogen bonds start to form to get us in our alpha helix beta and our beta sheet, okay? Then we've got our quaternary structure where our single polypeptide chain starts to bend round in its globular form as such. And then we go on to our quaternary structure where we have two or more proteins starting to work together, starting to kind of, should we say, embrace each other to become our functional protein. So some specific examples that you need to know, okay? So we've got a list of proteins here. Some of those you may have heard of. I'm sure you've heard of insulin. I'm sure you've heard of collagen, and you've probably heard of spider silk, okay? And you've got their functions there. So these are some specifics that you do need to know. All right, the human genome. So how does this all link together? Every human has a unique genome. So that's an entire set of genes that are individual to that specific organism. That's specific, we're talking humans, so that's specific human. Therefore, every human has a unique proteome. So it's an entire set of proteins in an individual. One gene can code for more than one protein. Which is more complex, do you think? The genome or the proteome? We'll discuss that further in class. What do you think? Okay, so we've got the genome, 20 to 25,000 genes, okay? Alternative promoters, alternative splicing, mRNA, we get our transcriptome, over 100,000 transcripts, post-translational modifications. Okay, so that can lead to a pretty high number of proteins in an organism. Okay, so our proteome is much more complex than our genome. Alrighty, so proteins. This is all going to become very, very important when we start looking at enzymes, but let me introduce to you this whole concept of the denaturization of proteins, denaturation of proteins. Okay, so on the left, you've got your protein in its native state. Quaternary structure, we've got 3 different proteins there, a pink one, a green one, and a blue one. If you look on the right-hand side, it's been denatured. The protein, so the quaternary and the tertiary structures have been broken down. Now, what can do that? So proteins can be denatured by heat and alterations in pH. There are a couple of other factors as well. Proteins function due to their specific shape. So if they become denatured, they can't function. Okay, so the shape, their actual shape is held together by chemical bonds. Remember, if those chemical bonds are broken, they become denatured, that protein cannot function. So having a look at temperature and denaturation. Okay, so we've got our protein there. You can see, I think there's 4 different coloured proteins there: dark blue, light blue, purple, and a pinky colour. And here it is, denatured. High temperatures break the hydrogen bonds. They don't break covalent bonds. It forces the protein back into its primary structure. It is denatured. Therefore, it is no longer a functional protein. Okay, here we've got a couple of graphs. I do love a good graph. We've got temperature increasing. I would say that is over time. And here we've got our protein structure. So a lovely quaternary structure there, breaking down there, even further broken down there. Okay, so as the temperatures increase, the protein denaturization is along. So it's gone from low denaturization to high denaturation over time. Here, okay, so we've got our temperature and our rate of a reaction. Now, this is where we start looking at our proteins as enzymes. So at this stage, we can see our enzyme is working at its optimal rate. However, as the temperature has increased further, our enzyme has stopped working. Its activity has started to drop until it's completely stopped. And that is because our enzyme is a protein and it's been denatured. Okay, we'll look further into enzymes coming up, so don't worry. Okay, pH and denaturation. Here we've got our protein. Okay, lovely. I can only see one colour protein there. Okay, it could just be the image. However, due to pH, you see here, it's saying that some of these denaturation agents are pH, temperature, also ionic strength, solubility. There are other factors that do cause denaturation, not just pH and temperature. But you see that the loss of the biological activity has occurred because it's become denatured. The presence of hydrogen and hydroxide interferes with the bonds of the protein. This changes the 3D shape. Sorry, these are ions, remember. This changes the 3D shape of the protein. Change in shape equals change in function. It is no longer active in the role that it was playing. Higher or lower pH than its optimal pH will both denature a protein. Lovely little graph again. Do love my graphs. This time again we're looking at enzyme activity because enzymes are proteins. Okay, and we've got its optimal pH here. If you decrease the pH, enzyme activity drops due to its denaturing. If you increase the pH, enzyme activity is dropping due to denaturing. The nature of science. Okay, on the left-hand side, you've got a table of the most common amino acids. There are 20 amino acids that are universally used in every living species on the earth. These are the 20 amino acids we learn about in biology classes all over the world. However, there's always a however, there are very few species that use an extra 2 amino acids and work with 22 amino acids to synthesize their proteins. These are rare and unusual, but they do exist. I feel some people may be doing a little research now. So that is the end of 2.4. Looking forward to covering some of those things again in class, just to really embed that knowledge. In terms of videos, I will see you soon in 2.5.